In this video, we're going to discuss one of the important fundamental process of cell, which is protein synthesis. I will cover some of the important steps that are involved in the protein formation. As you can see on the slide, I have tried to cover the eukaryotic protein synthesis, where activation, transcription, processing, as well as the translation steps are discussed in this video, and I will try to cover these topics in detail in my future videos. I hope that this video is going to help you to understand this particular process. And if you like watching these kind of videos, then please do hit the like button and subscribe to the channel. All right, let's start the video. Today our topic is the formation of protein from the DNA molecule. Here on the computer screen, you're seeing two structures. One is the nucleus and another one is the outer membrane of the cell. So let me write it down, the topic, which is protein formation from the DNA molecule. First, let's start within the nucleus. Inside the nucleus, you have the DNA molecule. And the step one will be the activation. In the activation step, what will happen? Transcription factor will bind to the DNA molecule, and this cause the initiation or the so-called activation of the transcription. Next, after the activation, you'll have the multiprotein initiation complex that will cause the formation of preRNA molecule or preRNA transcript. So you have these two steps. Let me write it down. Step one is basically the activation, where you have transcription factor binding. So this is also known as the regulatory part of the formation of the protein from the DNA molecule. So this is the regulatory part. And then you have the second step that we already discussed, which is transcription and the formation of a preRNA transcript, where you have that multi-protein initiation complex bind to the DNA molecule. You'll have RNA polymerase that will cause the formation of the preRNA transcript, and that preRNA transcript need to be processed. We are discussing eukaryotic transcription and translation, not the bacterial one. So everything is happening inside the nucleus. Here you will have that preRNA transcript, where you can see red regions and the blue regions. Red regions, they are basically the regions that are the coding regions, right? So the step three is the processing. In the processing, what you'll have, the removal of the non-coding regions. Here, non-coding region is the blue region. So blue region will be basically removed from that preRNA transcript. So you'll have, as you can see, I'm showing you the transcript where the blue region is getting removed, and you'll have mRNA molecule. mRNA molecule will further move to the cytosol. So that's the fourth step, where you have translation. That is the formation of protein. And this step is happening in the cytosol. So where you have these ribosomes molecules that are reading the genetic code and forming the protein sequence or the protein molecule of specific sequence. So in this step, you'll have formation of the protein from the RNA molecule. It is happening in the cytoplasm, and it involves the ribosomes. So this was the introduction of the protein formation from the DNA molecule. I hope now you understand what are these four steps. We have discussed activation, we have discussed transcription, processing, and the translation. So I hope that this video lecture is going to help you to understand this. In future videos, we're going to cover more detailed aspects of these different processes. I hope the video lecture was helpful for you to understand the process of protein formation. We have covered the basic steps that are involved in the protein formation process. I will cover these particular steps in more detail in my future videos. So if you like watching these kind of videos, then please do subscribe to the channel. And if you like the video, then please do hit the like button.